Hello guys, welcome to Arts Education, and I am going to discuss today about some multiple choice questions related to the genetic engineering. So, we will discuss one by one. So, first question is going to you. First discovered or isolated restriction endonuclease enzyme is. So, your options are BAMH1, EcoK15, or we can say Hind second. So, right answer will be here Hind second. Next question is going to you. In virulence apron of the agrobacterium, virulence dash gene act as a single standard binding protein. So your options are virulence A, virulence G, and virulence F, or that is virulence A. So right answer will be here by virulence A, which is acting as a single standard binding protein. Next question is going to you. BAMH1 enzyme is isolated from a bacterium. So your options are Bacillus thermus, Thermus aquaticus, Bacillus amyloliquefaciens, Bacillus pyrococcus. So right answer will be here, that is Bacillus amyloliquefaciens. Next question is going to you, that is size of human genome in base pairs. So your options are 2 into 10 to the power 9, 3 into 10 to the power 9, that is both of the above. Last one, the none of the above. So right answer will be here 3 into 10 to the power 9 base pairs. Next question is going to you, that is cDNA library is constructed by coding part of the DNA is called as. Your options are introns, B, exons, both, or none. So right answer will be here exons. Friends, we know that exons are the part which are the functional part, or we can say it codes for the different polypeptide chains, but introns are non-functional. So we have to remove in splicing from the mRNA. So next question is going to you. Hairy root disease in the plants caused by bacteria is known as. So your options are Agrobacterium tumefaciens, B, Agrobacterium rhizogenes, C, both, D, none. So right answer will be here Agrobacterium rhizogenes, which causes the hairy root disease in the plants. And we know that Agrobacterium tumefaciens, which causes the crown gall disease in the plants, or specifically in the dicot plants. So next question is going to you. Size of the ideal plasmid should be less than. Your options are 6 kV, 40 kV, 20 kV, 10 kV. So right answer will be here 10 kV. Next question is going to you. Range of the pressure, that is PSI, pound square inch, used in the ballistic gene gun delivery method. So your options are 200 to 300, B 100 to 200, C 300 to 400, D 500 to 700. So right answer will be here 500 to 700. Okay friends, next question is going to, that is tobacco mosaic virus are having type of the genome. So your options are single standard RNA, double standard RNA, double standard DNA, single standard DNA. So right answer will be here single standard DNA. Single standard RNA. Next question is going to you. Escherichia coli is a test type of bacteria which is act as suitable host for the gene cloning. Your options are gram negative, B gram positive, C both types, D none. So right answer will be here gram negative bacteria. Friends, we know that the bacteria is classified as the two types, that is gram negative as well as the gram negative positive. Next question is going to you. What is the size of PVR 322 vector? Your options are A 4.5 kV, B 4.3 kV, C 6 kV, 5.8 kV. So right answer will be here 4.3 kV. Friends, we know actual size is the 436 and 3 base pair. So we can see here, we can write here 4.3 kV. Next question is going to you. Which of the technique is used as a physical method to transfer the gene into the host? So your options are calcium phosphate method, B microinjection method, C both, or D gel electrophoresis. So we know that already, that is calcium phosphate method is the, that is chemical method, because these are the chemicals which are used in the to transfer the gene into the host or in vector, and B microinjection, this is the physical method. So right answer will be here physical method, that is microinjection. Next question is going to you. It is required to distinguish between the cells that have taken up the vector and that have not. It is done by using your options are multiple cloning sites, B array of replication, C high copy number, D selectable marker. So friends, to detect them that is it is vector transferred or not, if because vector or plasmid having the selectable marker, we can see that is antibiotic resistant genes like the ampicillin or tetracycline. So on the basis of that, we can select our plasmid that is transformed or not transformed. Next question is going to you. Which of the statement is true for the PVR 322? Your options are A. It contains only an ampicillin resistant gene. B. It contains both ampicillin resistance and plus the tetracycline resistant genes. C. The cloning site is present only in the ampicillin resistant gene. D. It is a natural factor. So right answer will be here. That is, it contains both types of the selectable marker and ampicillin resistance as well as the tetracycline resistance gene. Next question is going to you. If high copy number is there, the replication is called as. If low copy number is there, replication is called as. So if any plasmid factor is producing high number of the copies, we can say large number of the copies. So it is called as the stringent type. And if any plasmid factor which is producing low number copy, so it is called as the relaxed type. So right answer will be here A, that is stringent as well as the relaxed. Next question is going to you. Which types of the ends are produced by EcoR1 during cleavage of the recognition sequence? So there are the two types of the ends produced by the different bacteria. We can say that is staggered end or blunt end. So staggered end are the two types, 5' producing ends or 3' producing ends. So here EcoR1 is producing 5' producing ends. So right answer will be here A, that is 5' producing ends. Next question is going to you. Which enzyme is used in to remove 5' phosphate from the DNA ends? Your options are polynucleotide kinase, alkaline phosphatase, C reverse transcriptase, D S1 nuclease. So right answer will be here alkaline phosphatase. Friends, generally two terms are there, that is phosphatase or kinase. So kinase always working, that is addition of the 5' phosphate, and phosphatase always working to remove the 5' phosphates. Okay, next question is going to you. Which of the plasmids contain that a minimum of 250 base pair of the lambda DNA? That is your options are plasmid, B cosmids, C plasmid or phasmid. So right answer will be here cosmids. Next question is going to you. Which of the following is a expression factor? Your options are phasmid, B cosmid, C both, D none. So right answer will be here phasmid. Friends, why we are calling it a phasmid is an expression factor? Because any gene which it wants to express, it requires like a promoter as well as operator structural genes which are present in the phasmid factor. So we are calling it is expression factor. Next question is going to you. The vector which is requires two hosts, two different species to replicate is called as VAC. We can say vectorial artificial chromosome. B YAC, yeast artificial chromosome. C shuttle vector. D none. So right answer will be here shuttle vector. So friends, these are the questions which are related to the genetic engineering, or we can say RDT, the recombinant DNA technology. So I think this video is helpful for you. Thank you so much.